In this video, we are going to be concluding our lecture on the anatomy of the excretory system. In the previous video, we have described the renal pyramid, or known as the renal medulla, right? And I've described two types of nephron that we're having. I said we have two types of nephron based on the length of their loop of Henle. We have the cortical nephron and the juxtaglomerular nephron. And I said juxtaglomerular nephron means the one whose glomerulus is closer to the medulla, which is this one here. This is the medulla, this is the cortex, and the glomerulus is what is closer to the medulla than the cortex. So that's why I call it juxtaglomerular nephron. And we also show that the blood enters into the The blood enters into the glomerulus through the afferent arteriole, right? Through the afferent arteriole, yeah, and leaves through the afferent arteriole. We mentioned this one to be the renal corpuscle, and we mentioned this ones to be the vein, the blue stuff here. We mentioned them to be the vein. Now, I have a slide here that shows the passage of passage of blood into and outside of the kidney. And the first one that I described that day was the entrance of blood through the renal artery, then from the renal artery to the segmental artery, from the segmental artery to the interlobar artery, which is the artery between lobes, right? These are the arteries between interlobar arteries, right? And this is also interlobar artery, this is also interlobar artery. And I said interlobar artery branches to form the arcuate artery. The arcuate artery branch to form the cortical radiated artery, also known as interlobular artery. You understand? And I said we have two arteries. We have arterioles in the cortical radiated artery. We have arterioles, and we have two types of arteriole. We have the afferent arteriole, which brings the blood into the glomerulus, and the efferent arteriole, which brings the blood outside of the outside of the glomerulus. So after blood has been filtered, it is going to be going outside of the outside of the glomerulus through the efferent arteriole. You understand? And we have the from the efferent arteriole is going to two types of arteries. Either it is peritubular capillaries or the vasa recta. The vasa recta is the one that is are those arteries in the medulla. You understand? The ones that extend into the medulla. You understand? While the peritubular capillaries are those ones that stays on the cortex. So both of them serve the same purpose. Some blood will enter into the vasa recta, some will enter into the into the peritubular capillary. The purpose of them is to deposit the blood, the filtered the filtered blood into the into the interlobular vein, also known as cortical radiated vein. Then from there to the arcuate vein, from the arcuate vein to the interlobular vein, then to the renal vein outside of the outside of the kidney. So that is just the process that I explained in the last lecture. Now, let us now talk about the glomerulus itself. Though there are still some things that I need to mention in the nephron, like the loop of Henle, which I haven't explained that much, and the function of the thin limb descending, the thin descending limb and the thin ascending limb, the thick ascending limb as well. I haven't mentioned these ones, but for now, let us just talk about the glomerulus itself. Remember I said the glomerulus is found in the Is found in the renal corpuscle. And the renal corpuscle consists of two things. It consists of the let me zoom that in very well. So look at this. It consists of the It consists of the Bowman capsule, right? And tuft of capillaries, also known as the glomerulus. You understand? So look at it. These are capillaries network, also known as Also known as the glomerulus. Now, the glomerulus now consists of Consists of blood capillaries and filtering units. You understand? We can call the glomerulus the filtration unit of or the site of filtration of blood. You understand? So Bowman capsules surround the glomerular capillaries. You understand? And it is composed of epithelial cells. You understand? Look at this. The epithelial cells around this place. You understand? The epithelial cells surrounded by epithelial cells, and it contains of It contains of filtered blood, also known as glomerular filtrates. You understand? Now, we can also find some cells inside the glomerulus. Example of cells that we can find in the glomerulus is mesangial cells. We can find mesangial cells inside the glomerulus, and also we can find something. We have the podocyte as well as the endothelial cells. We'll be talking about that later. You understand? So the outer surface of the glomerulus is termed in glomerular capsule. Let me change this one to black. Now, the outer space is called the what? It's called the glomerular capsule. The space between the glomerular capsule and the glomerulus itself is called the what? Is called the capsular space. You understand? This is the capsular space. We have the juxtaglomerular apparatus, which I will be talking about later. We have the granular cells, also known as juxtaglomerular cells. We have the densa macula, or macula densa rather, macula densa. We have the podocyte. We have the glomerulus itself. This yellow stuff, the one that around here is the glomerulus, and this one here is the proximal convoluted tubule. If you notice very well, there is microvilli here. The function of this microvilli is to increase the surface area of the proximal convoluted tubule to ensure what? To ensure absorption. You understand? So most of the absorption occur in the proximal convoluted tubule. Even 75% of the reabsorption occurs in the proximal convoluted tubule. You understand? The other one occurs in the loop of Henle as well as the distal convoluted tubule. So 75% occurs in the what? 75% occurs in the proximal convoluted tubule. Now, remember I told that we have two arterioles. We have the afferent arteriole that brings the blood inside the what? Inside the glomerulus. And we have the efferent arteriole that brings the blood outside of the glomerulus. Now, the force, there is no how you're going to have the flow of blood that there won't be a force that is pushing it, you understand? So there is a force that determines the what? That determines the flow of blood out of the glomerular capillaries. We call it Starling force. Starling force. Starling. You call it Starling force. And also, there is two pressure. That is hydrostatic pressure and the oncotic pressure. The hydrostatic pressure is found in the capillary and the oncotic pressure is found in the Bowman space. They push the fluid out of the capillary into the Bowman space. So we have the Bowman space somewhere around here. You understand? The Bowman space somewhere around there. That is the space between the what? Between the capsule and the proximal convoluted tubule. You understand? Or should I say the space between the capsule and the capillaries. That is the capillary network of the glomerulus. That is called the Bowman space. So the hydrostatic pressure and the oncotic pressure push the fluid out of these capillaries, out of this particular capillary that is formed by the afferent arteriole, into the what? Into the Bowman space. You understand? Then into the proximal convoluted tubule. Now, the hydrostatic pressure in the Bowman space and the oncotic pressure in the capillary draw the fluid from the Bowman space into the capillary. That is, if it wants to go back. Remember, I told you that the stuff is going to the blood is coming from here. It is going to be filtered around this side. Can you see? It's going to be filtered around this side. Then it's going to be deposited into the Bowman space. You understand? It's going to be deposited into the Bowman space. The ones that is sieve will be going into the proximal convoluted tubule, and the ones that are good is going back into the capillary. You understand? So the hydrostatic pressure and the oncotic pressure will determine the what? The flow of blood back into the capillary. You understand? Now, at this point, the hydrostatic pressure and the oncotic pressure in the Bowman space, that is, the hydrostatic pressure here and the oncotic pressure that is around here, is significantly lower than the one inside the capillaries. You understand? Now, if you notice very well, if I zoom it in very well, you will see all these dash. Can you see? Those are the podocytes. You understand? Those are the podocytes. Those are the filtering units, which I will be talking about later. You understand? Look at this blue stuff. That is the podocyte cell body, or let me call it the nucleus of the podocytes. You understand? Now, as the blood flows through the capillaries, the hydrostatic pressure decrease. The hydrostatic pressure here decrease until it is almost equal to the oncotic pressure. You understand? Until it's somewhat equal to the oncotic pressure. And the function of the hydrostatic pressure, in the capillaries here is determined by the afferent arteriole and the efferent arteriole. You understand? The afferent arteriole and the efferent arteriole. Once the afferent arteriole dilates, the hydrostatic pressure is going to be high. It means once this one dilates, this one is going to constrict, so that no blood will flow outside, only blood will be flowing inside. So this one is going to dilate, this one is going to constrict. Once this one dilates, it is showing that there are blood that are coming inside. Increase your pressure. You understand? So the pressure is going to increase in the capillaries. You understand? As the blood is flowing inside, and this one is going to constrict. So once this one dilates, this one is going to constrict. You understand? This afferent artery is going to constrict, and once it constricts, the hydrostatic pressure will what? the hydrostatic pressure will be hydrostatic pressure will be lower. You understand? Remember, we call it feedback mechanism. It's just like feedback mechanism. It's just like is it positive? I think positive feedback mechanism. As this one dilates, the hydrostatic pressure increases. As this yeah, and as this one dilates, this one is going to constrict. You understand? So it's a feedback mechanism. It's a form of feedback mechanism. Now the now remember I told that we have something around this place. We call it podocytes, right? We call it podocytes. So what are podocytes? Podocyte means podo, from the word podo. Let me write that down. From the word podo and site. Site. Podocyte. So it means foot cell, you understand? Or should I said pedicell. Pedicell. Cell. That is cell with many pedicels. You understand? Like cell with many pedicels. Or let me say multiple feet. So multiple feet. They have multiple feet. I'm going to be showing you what that means now. Multiple feet. You understand? Now, this is an example of what a podocyte. This is a podocyte. You understand? This is the structure of a podocyte. Now, this, can you see? We have pedicels. Can you see? These are pedicels. Somewhere around here. Let me clean that one and use arrow. So these are pedicels. You understand? Because this is a pedicel, this is a pedicel. And we have filtration sheets somewhere around here. You understand? So these podocytes, they form two types of processes. They form the primary process and the secondary process. They form the what? The primary process and the secondary process. But the primary process is always bigger than the secondary process. You understand? Which means the primary process is small and the primary process is big, secondary process is what? Is small. And the both of them interdigitate with one another, you understand, and leave a very small space between adjacent cells called the slit pores, you understand, something like this, or this pores, you understand, called the what? Called the slit pores. So we have the outside of it to be capillary lumen and the inside to be glomerular capillaries, you understand. So these are the pores that they use for what? That they use for filtration, you understand. Now, it should be noted that all the capillaries inside the glomerulus, all the glomerular capillary, they are fenestrated. And when we say something is fenestrated, fenestrated capillaries are required when there is a lot of exchange between substances. There is a lot of exchange of substances between blood and interstitial tissue. You understand? So all capillary in the glomerulus or all glomerular capillaries are fenestrated capillaries. You understand? They are large enough to allow filtration of components, you understand, except the blood cells. Are you getting me now? They are large enough to what? To allow the filtration of components except the blood cells. So the second part of filtration membrane is the endothelial cells. Let me go back. Endothelial what? Endothelial cells. That is endothelium of fenestrated capillaries. That is the second what? That is the second filtration membrane. The endothelial cells prevent the filtration of large protein. It prevents the what? Filtration of large protein molecule. And the third part is the slit pores. You understand? The slit pores. Look at it. The slit pores. You know, I've mentioned the basement membrane of the capillary, the endothelial fenestrated cells, and the slit pores. And the function of the slit pores is that they produce slits, which prevents the filtration of medium-sized protein. So the first one prevents the escape of blood cells or filtration of blood cells. The second one prevents the filtration of large protein, and the third one prevents the filtration of what? Of small-sized protein. So these three structures constitute the filtration membrane. They constitute the what? The filtration membrane. Now, let me just say glomerular filter. We have glomerular filter, that is GF, will now be equals to fenestrated endothelium plus basement membrane plus slit pores. So this one is glomerular filter equals to fenestrated endothelium, basement membrane, and what? And slit pores. You understand? That is Fe plus Bm plus Sp. So that will make you remember. You understand? So these are the what? These are the components of glomerular filter, also known as the podocyte. It should be noted that this place, look at it. This is the podocyte cell body. This is the podocyte cell body, and this is the nucleus of the podocyte. Remember, it's a cell. It's going to possess a nucleus. You understand? Now, these podocytes, they have remember I told they interdigitate with one another. There is now a protein-based structure known as slit diaphragm. You understand? And this slit diaphragm, it consists of a primary protein component known as nephrin. It consists of a primary protein component known as what? Known as nephrin. And this protein component is the primary component of what? Of the slit diaphragm. Are you getting me now? So that is just the basics of the what of the podocyte itself. You understand? So basically, podocyte forms the what? Forms the filtration membrane of the what? Of the glomerulus, of the glomerular capillaries. You understand? So moving from there to denser. Now, remember that I mentioned that we have something like juxtaglomerular apparatus. Now, juxtaglomerular apparatus is a specialized structure that is formed by the distal convoluted tubule and the glomerular afferent arteriole that is near the vascular pole of the renal corpuscle. I repeat, it's a specialized structure that is formed by the distal convoluted tubule and the glomerular afferent arteriole near the vascular pole. You understand? Now, there are two basic functions of this complex, this juxtaglomerular apparatus. There are two basic functions. The first one is the systemic blood pressure. It regulates what? Systemic blood pressure. So it regulates pressure, blood pressure, uh, blood pressure by secreting an enzyme known as what? Known as renin. So they secrete what? They secrete renin in order to what? In order to control the or to regulate the blood pressure. You understand? And this renin is part of the renin angiotensin aldosterone system. Renin angiotensin aldosterone system. That is our renin angiotensin aldosterone system. You understand? So these the second function is to control the filtration rate of to control the filtration rates of the renal corpuscle. The filtration rate of the renal corpuscle. And how do they do this? They control the amount of blood that flows through the afferent arteriole. That is what they do. So the juxtaglomerular apparatus consists of three apparatus, like three basic cells. The first one is macula densa. Macula densa. The second one is JG cell, that is juxtaglomerular cells. The juxtaglomerular cells. And the third one is mesangial cells or extraglomerular mesangial cells. Extraglomerular mesangial cells. So these are the cells that made up the what? That made up the juxtaglomerular apparatus. You understand? Juxtaglomerular apparatus. So this is look at this. Let me zoom it in well. So this one is the juxtaglomerular cells. You understand? This is the juxtaglomerular cells. The reason why they call it juxtaglomerular is because it is close to the glomerulus. Juxta means closer. So it is close to the glomerulus. That's why they call it juxtaglomerular cells. You understand? And this is the denser, the macula densa. This is the macula densa. You understand? And the macula densa, what does it do? It has a sense that sense the sodium chloride concentration. You understand? So it is an indicator for glomerular filtration rates in the nephron. So that is the site of it's just that's just the basic function of it. You understand? So after the filtration has happens, what happened is that the filtrate, we call it filtrate, not urine yet. The filtrate is going to what? Is going into the what? Into the proximal convoluted tubule. And I told you that 95%, 75% rather, 75% of the reabsorption occurs in the what? Occurs in the proximal convoluted tubule. And the reason why it occurs there is because of the what? Of the brush border. You understand? They possess something like microvilli at the what? At the base. You understand? They call it brush border. You understand? At the filtration. So they have brush border at the barrier. You understand? So most of the absorption occurs in the what? Occurs in the proximal convoluted tubule. So majority of glucose, amino acid, ions, water, and even protein you understand, will be reabsorbed in the what? In the proximal convoluted tubule before they move to what? Before they move to loop of Henle. Now, look at it. This is the loop of Henle. Let me show. This is the loop of Henle. Let me just show this. The loop of Henle. So in this loop of Henle, we have the thin limb descending loop of Henle. That is thin limb descending loop of Henle. Thin limb descending loop of Henle. We have the thin limb ascending loop of Henle, and we have the thick limb ascending loop of Henle. Now this thin limb descending loop of Henle, the cells that are there are squamous epithelium. Squamous what? Squamous epithelium. And this squamous epithelium is permeable to water but impermeable to sodium chloride. So the function of loop of Henle is two. You understand? The first one is absorption of water. The second one is the absorption of the salts. You understand? The salts. That is the reason why around this medulla, there is higher osmomolality due to what? Due to the absorption of salt by the loop of Henle. So this place, you understand, this place, and this place, basically they absorb water and what? Water and salt. So the concentration of sodium chloride around this place is higher, you understand. So they increase the osmomolarity of the interstitium by removing the what? By removing sodium chloride from the what? From the filtrates. You understand? They remove sodium chloride from the filtrates. Are you getting me now? Now, the thin descending limb absorb water, or let me say the absorption of water occurs in the what? In the thin descending limb. But the absorption of sodium occurs in the thin ascending limb. It also consists of squamous epithelium, you understand? And the thick ascending limb consists of cuboidal epithelium. Cuboidal epithelium. It is also impermeable to water, but it actively absorbs sodium, you understand? So look at it. Water is absorbed here, salt is absorbed here, salt is absorbed here. Can you see that more salt is absorbed than water in the what? In the medulla, you understand? This is the medulla, you understand? So there is higher osmomolarity around this what? Around this place, you understand? So that is the description of the what? Of the glomerulus and the loop of Henle. Remember that after it moves from the loop of Henle, it is going to where? It is going to distal convoluted tubule, you understand? And this distal convoluted tubule, it lacks brush border. You know, proximal convoluted tubule has brush border or microvilli. So this one doesn't have microvilli. The distal convoluted tubule doesn't have what? Doesn't have microvilli. So this is an example of distal convoluted tubule. You understand? This is distal convoluted tubule. It doesn't have microvilli, and it now deposits into the what? Into the collecting duct. So the collecting duct, it performs two functions. It performs what? It performs two functions because they have two types of cells. They have the intercalated cells and the principal cells. Those are the two cells that are present in the what? In the collecting duct. You understand? And the intercalated cells or intercalated cells, they regulate the pH by the addition of H plus or HCO minus. So the addition of hydrogen ion occurs in the what? Occurs in the collecting duct, which make the urine to be acidic, because once it enters the distal convoluted tubule into the collecting duct, we can now call it urine. You understand? And the principal cells control the it regulates the solute concentration by absorption of water, by regulation of absorption of water. And this principal cell is under the control of antidiuretic hormone, ADH. Antidiuretic hormone, you understand? It is under the under the control of antidiuretic hormone. Are you getting me now? And it either increases what we know as aquaporin, or it maintains the aquaporin. We call it aquaporin. You understand? So from the collecting duct, the absorption of water occurs and stuff. Then it goes into the what? Into the renal papillae. Into the renal papillae. You understand? Then from the renal papillae, it is going into the microcalyx. The microcalyx. So from the microcalyx, it is going into the major calyx. You understand? The minor calyx, into the major calyx. And from the major calyx into the what? Into the pelvis. Into the pelvis. Are you getting me now? Then the pelvis now forms ureter. You understand? Now the ureter, what does the ureter do? It connects the kidney. It connects to the kidney, to the urinary bladder. But one thing you have to note that the ureter is a modular tube. Can you see? It is like a tube. Look at it. So it is modular too, you understand? And it consists of longitudinal layer, that is inner longitudinal layer, and the outer circular layer. Inner longitudinal layer and the outer circular layer. And the type of cell that is here is transitional epithelium. What do I call it? Transitional. And you know when this is something is transitional, it means it can change. Transitional epithelium. So it can stretch. Are you getting me now? Transitional epithelium. It can stretch, which means it can relax, it can dilate. It can dilate, it can constrict. You understand? Then it is now passed into the bladder. Are you getting me now? Into the bladder. Then from the bladder to the urethra. Then it is passed outside of the body. But you have to know that the macula densa, I say macula densa, the major calyx, the minor calyx, the major calyx, the pelvis, the ureter, the ureter, the urinary bladder, and some part of the urethra. All of them consist of transitional epithelium. That's the reason why they can contract and they can relax. You understand? So they consist of what we know as transitional epithelial cells. You understand? So and these so this is it. So from the ureter, to the urinary bladder, then from the urinary bladder to the what? To the urethra. Then it is passed outside of the body. Remember that the cells here is transitional, the cell here is transitional, and the cell here, some of the cells here are transitional. You understand? Some of the cells here, they are transitional. So that's on the anatomy of the urinary system. Thank you all for listening. We really appreciate, and subscribe for more. If you have any question, you can just drop it in the comment section. Thank you all for listening.